Let's pick up HIV entry and let's talk about the envelope protein. So I'm showing a Vimeo video that's actually pretty decent up here. Leaves out a couple things, but it's all right. Just Google Vimeo HIV entry or HIV fusion process. So here is the envelope protein. Let's look at its parts. We want to start with the overview and then we'll go through the things that it's going to go through. We'll try to keep them in chronological order as far as we can, but there are certain things that may be happening at the same time. All right, so here's the organization. There is a signal peptide. Now the signal peptide is going to be cut off by signal peptidase, and that's going to happen very early. That happens before we even make the virion that is now going to be entering. So I'm showing it there in gray because it's like a little ghost. It doesn't exist anymore. Then we have some of the parts of GP120. So GP120 is going to be the big thing that is in blue right here. And we're going to have our variable loops, the V1, V2, because they're very close together, as you can see in the alignment video. And the V3 loop, which is going to be very important for co-receptor binding, is down here. V4 and V5 would be in this region, and they don't do anything particularly interesting that we know about or that I can talk about, so forget about them. Each of these little pitchfork things, like this or like that, is a glycosylation site. So you can see there are glycosylation sites on the V1, V2 loop, and there are glycosylation sites on the V3 loop. Now normally V1, V2, and V3 are going to be tucked away, or rather V1 and V2 are going to be sitting on top of V3 and blocking access to it, so that if you were an antibody, for example, you would have a hard time getting to the V3 loop until later, until it comes out. Okay. The beta 20 and beta 21 is it's a little beta strand, and it is important. This is another one of these conformational switches. We're not going to talk about it later. If you want to Google it, that would be a good thing to add to your answer on entry. But it's a thing that's going to change as we go through the process of HIV entry. There's a lot of parts of this structure that are going to change. And you can think of the whole thing as being something like spring-loaded. There are a lot of things that can happen when other constraints are lifted. Like that. There you go. All right. So down here we have the fusion peptide. That's the FP. Heptide repeats 1 and 2. And there's a little glycosylated region in between those two. The MPER, the transmembrane, and the cytoplasmic carboxyl terminal tail, which is actually going to form this sort of flat disc with two myristates on each one of the 3 monomers. And that is going to basically lock the bottom, the tail end of envelope protein into the bottom interface of the membrane. All right. And this is going to be cleaved. The activation cleavage is going to be here. There's actually 2 cleavages. They're both done by a furin-like protease, which is a family of different proteases. There are a bunch of them you would find in the lungs. There are a bunch of them you would find on macrophages. There are a bunch of them you'd find in the blood, wherever else. Wherever HIV is, there's going to be a furin-like protease, and it's going to be able to cut these things. That cleavage is going to separate GP120 from GP41, and we need that for later. And it's going to separate it at the fusion peptide. The fusion peptide is the first thing after the separation. So it's necessary to free up the fusion peptide. There are going to be 2 furin-like cleavages possible. The first one is optional. The second one is mandatory because the second one is right at the fusion peptide. And we'll get to that. We got a slide about that later. The other thing to look at is this 501 and this 605 right here. This is a disulfide linkage. And so this disulfide linkage is going to hold GP120 and GP41 together even after furin cleavage, so that the furin cleavage doesn't just make the GP120 fall off immediately. That means furin cleavage can happen at a range of times. It can happen really early to really late. Oh, look at that. We're budding our new virus. That's beautiful. And they did a great job with the gags there. But that's for later. We'll rewind this video and watch it again here in a sec. All right, let's go. So we'll start with the furin cleavage. There's a range of time when this can happen. It just has to happen before we are in the endosome and it's acidified and we're going to make the conformational change. the final conformational change that exposes the fusion peptide and then forms the 6-helix bundle. That'll make sense in a minute. I'm just going through this now so that when you rewatch it, then you can say, oh, that's when it happens. So furin is going to cleave after basic amino acids. The two most basic amino acids are going to be K and R, and by basic I mean they can take on a positive charge at fairly normal pHs that you can reach. And so these are going to have positively charged side chains rather on the amino acid. And after that second R right there, and after this KR here of the REKR motif, that's where your cleavages are going to be. And so this is the first one, and the second one, if it cleaves here, that is going to be cleavage at the site of the fusion peptide. There we go. At the end terminus, like beginning of the fusion peptide. All right. Let us continue. And so you can play with these. You can change one basic amino acid to another one. It'll still work. You can knock out the first one. The second one will still do the job. But if the first one's there and gets cleaved, then it makes it easier to cleave the second one. It sort of frees it up. What's going to happen is you're going to get this little, there you go. That's the fusion peptide, and it's able to sort of flap around out there once it's free. Because it starts out with a fusion peptide as just like the middle of the envelope protein. So you got your your GP41, your GP120 over here, and when it gets cut, then the fusion peptide can flap around, and then it can start to do conformational changes that it needs to do for later. So this is just an NMR structure showing the different conformations that are most common for the fusion peptide, but you can see they're all different and they're all sort of flopping around. That's what I mean. Yeah, these are 4, let's say, equally likely possibilities, or 5. The pink and the orange are pretty close to each other. Okay, so first of all, when we get to this cell, the initial point of contact that we actually know about that's like specific is going to be contact between CD4 and GP120. CD4 has a phenylalanine at position 43. This is F43, F for phenylalanine. That's the single letter amino acid code. It is actually spelled with a PH that makes an F sound. That's going to stick out like a little thumb on the top of domain 1 of CD4, and there's going to be a little hydrophobic pocket that's going to bind it. The hydrophobic pocket is what's shown in yellow, pink, and red over here, and there are drugs that will mimic the binding, and those drugs will, nope, let's go back. Yeah, we like that. All right, there we go. Sorry, gotta tend my video. Those drugs can prevent CD4 from interacting with GP120, which is the first step of the process and starts all the other stuff off. So these are the names of 2 of the drugs that happen to work, and yeah, that's fine. It's more to show you the little star. The little star shows you where phenylalanine 43 fits into this pocket. We can zoom in. This is actually from the bottom of the same, or no, the top of the same diagram as the one above. So same paper. This is showing you the 2 drugs and how they reach into the pocket. Now, there are some structures of phenylalanine 43 being in that pocket, but they're not as clearly depicted as this, so I like this one. See the ring there? That's the ring of the drug, and it's mimicking the ring of the phenylalanine, because it's one of those with the aromatic ring in there. And so, yeah, basically that's how you bind in the pocket. You have a phenylalanine-shaped molecule that is able to interact with the pocket and form the right sorts of hydrogen bonds and maybe a pi bond down there to stabilize it. Okay, so that's the pocket, and just imagine this black thing is like we've sliced through the glycoprotein, and so you can see inside of it. The gray thing is like the back of the pocket. It's like we've cut through this big solid thing, and you can see the pocket shining through. After CD4 binding, or upon CD4 binding, so we add CD4 to the mix. So this over here is GP120. And GP120 has a couple different subdomains, if you like, or domains. That's just like a part of a protein that does a job. This is the inner part, I believe, over here. This is the outer part. And those were shown on a previous diagram back here. There we go. as layer 1, the inside, and layer 2, the outside. There you go. So you can see the V1, V2 are sitting with layer 2 on the outside, basically. And so there are two little beta sheets, little tiny ones with just 2 strands each, and that's these pink things that are over here and here. Those are normally separated. So one of them hangs out with the inner face, and the other one hangs out with the outer face. When CD4 binds, it's actually going to rotate those molecules around. That's going to bring these two things into contact, and they're going to form a single 4-membered beta sheet. And that beta sheet is going to be called the bridging sheet. It's locking together these two parts, the inner and outer parts, and this is going to help GP120 clamp down on CD4 so that it doesn't just, lose its grip on that one little phenylalanine. That's not much to hold on by. It's like holding on to a little pinky finger like that. And GP120 is going to get a massive, like baseball mitt grip around this CD4 protein. The thing it's grasping right here, the thing they're showing, is just domain 1. This is what's called an immunoglobulin-like domain, domain 1 of CD4. And there are 4 of these immunoglobulin-like domains that make up CD4. You could think of it as CD4. Oh, it has 4 immunoglobulin-like domains. Yeah. And occasionally those things work out, the numbers match up, and just as often they don't, but in this case they do. So whatever, remember it that way. Remember it another way. I'm not your mother, yeah. I'm not your father either, yeah. Let's see. Here is a summary of a bunch of the bigger, more important changes that we know about that happen when CD4 binds to GP120, or rather, it's like that goes in a pocket and then bam, we're going to clamp those 2 domains, sort of rotate them around and lock it in, lock tight. So the first change, let's just think structurally, you go from a sort of a closed conformation like this to an open one. Closed before, after binding, open. So you could say that the binding is going to activate and open this. And so that's the big change in the structure. And so you can see that the green parts are together, and here the green parts are apart. And there's a big old thing in here. So each of these domains, like the inner domain is now clamped to the outer domain, and they're pinching onto a CD4. What you're going to see is this is going to be closed, and then these 4 are going to be open, but this is the least open one of those 4. So sometimes this will look like closed, sometimes open. It's like a partial one. This is what we get. These 3 images are going to be what we get, the structures of what we get when CD4 is all the way bound. This is like the end goal. So, starting out, V1, V2 loop is going to be covering over the V3 loop. V3 can't get out because V12 has to get out of the way. And after CD4 binding, bam, look, V12 is going to point down. V3 can now flap around up. None of these things are like rigidly held in these positions, but they're flexible and they're able to move. And so think of it as an approximate position, if you like. Next thing that happens is going to be, this is just showing once again the V3 loop is in there and it's covered. V12 loop is the green bit, and it's covering over. V3 is this little thing that's underneath and it can't get out. Oh, it's trapped in there. And now after binding, V12 is going to get pulled over, and it's going to be interacting with, as part of that clamp that's holding on to phenylalanine 43. And that means V3 is free to flop out and hang out and do its thing, binding to the coreceptor. Next thing, we've got our bridging sheet. So we have 2 of these 2-strand helices. One of them actually has a 3rd strand that sort of sits next to it that's not going to participate further. And these things are going to come together. So this is our 2-strand, here's our 3-strand intermediate and the partly open one. And this is going to change to a 4-strand big sheet by bringing those 2-stranders together to make a 4-strand sheet. Yeah. And so that's going to be the open conformation, is going to have the bridging sheet. And the bridging sheet is going to, it's like a clamping sheet, really. It's going to lock those 2 domains together and form a bridge between the inner and outer parts of GP120. This is the same as that. I just copied it over so you can read the columns underneath. The next change is that the fusion peptide is this little squiggly pink bit, squiggling around right there. It is not able to form an alpha helix in the pre-fusion closed conformation. But in the open conformation, the fully open conformations anyway, 1, 2, 3, you can see it turns into that little pink squiggle, and it's attached there. Here's the little bend in it. This is the hook-shaped fusion peptide, is able to assume the fusion peptide conformation. Now, if this thing is still not been activated by furin cleavage, then that would be another impediment. It would be harder for this to fold up into the alpha helix that it needs to be, because it would still be attached to the rest of GP120. Yeah, so that wouldn't work. The next change is going to be down in the transmembrane part. So this is going to be in GP41. This is heptad repeat number 1. So heptad repeat number 1. At the C terminus of this, so the far end, bottom end of this, there's this little bent gold helix that's not interacting. And when we activate these, it's still not going to interact, but it gets longer. It goes from a little helix to, look at this big long helix. So we're going to extend that, and this bit at the top that was not an alpha helix to begin with is going to extend and is going to interact. So we're going to form additional interactions that hold the 3 heptad repeat 1s together at the center, at the core of GP41. And they're going to stay together. Those 3 stay together, and the heptad repeat 2s are going to bind on the outside of it, just like that, later on in the fusion process. The last thing that happens, and this is just like a bookkeeping thing, is you get this region that is not an alpha helix. They call it alpha 0 because it's not an alpha helix, is going to turn into an alpha helix. That is another marker of CD4 binding, and it's like a decent sized structural change. Each of these changes changes the shape of CD4. Antibodies that can stop a virus from entering recognize shape. Each time the shape changes, that changes the availability of that part of the virion to bind, or that part of the envelope protein to bind with antibodies. So you will have antibodies that can only bind to certain conformations, and those conformations are going to change. It's like they're the Terminator, the Terminator 2 Terminator, this liquid metal that can just morph into different shapes. Since antibodies work by recognizing a shape, changing shapes makes you innately a little, a hard target, harder to hit. The other thing that's a problem here is that the closed conformation is going to be the one that your immune system would really like to be able to recognize. Because if you recognize that, you don't get infected, because you can stop the virus before it does these things. But most vaccine versions of the envelope protein, you can stabilize it in the open conformation like this, or the half open conformation. It's real hard to stabilize in this conformation. And this one is just not as immunogenic. It's harder to get those neutralizing antibodies, because you really want all the V1-2 loops to be, clustered together there like a little crown on top, so that you can lock those down with your antibodies and, make it work. So, all right. After CD4 binding, after all those conformational changes, and each one of those conformational changes is a little step down in terms of energy. So each one of those is going to be more favorable once it's made the change, but there's like an activation energy, like in a chemical reaction. Unless you have CD4 there, which is causing these domains to rotate around and stick together and bind to that phenylalanine, there's no way you don't have the activation energy to get these other changes to happen. So it's like a, step 1, then 2, then 3, then 4, and imagine all those changes are happening at around the same time after CD4 binds, or because CD4 binds. Next thing that happens is going to be the extension of the V3 loop. The V3 loop is this thing right here. It has an alpha helix on it. The alpha helix extends down into the pocket of the co-receptor. If this is a macrophage, the co-receptor is probably going to be CCR5. If it is a helper T cell, the co-receptor is probably going to be CXCR4, although the immune system has got all kinds of variation in there, and my tiny brain can only understand so much of it. So there are going to be cases where you would have different types of cells, or the wrong co-receptor potentially. Yeah, go ask an immunologist. They'll tell you all about it. They love this stuff. The V3 loop is mimicking the shape and the hydrogen bonding of the natural ligand. So normally we would bind to the tail end of CCl5. That's what CCR5 is going to bind. And it makes a little alpha helix down here and interacts with the pocket. Well, V3 loop makes a little alpha helix and interacts with the pocket in roughly the same way. And so that's how HIV is going to get in. It's like a little Trojan horse. It's making it look like, oh, I'm the normal thing that you should be binding, CCR5 or CXCR4. So you should bind me. And it does, yeah. And this can only happen efficiently after V1 and V2 have moved out of the way, which is after CD4 binding. Of course, right? You've got this. You know this. Okay, then you will get a conformational change to these co-receptor. The conformational change is going to tilt some of these helices in by just a little bit, but it's enough that they can get a real nice grip on the V3 loop or CCL5. That's how G protein coupled receptors work. And CCR5 and CXCR4 are both G protein coupled receptors, which means that off of this helix 7 over here, there's going to be a G protein coupled to the receptor. This is the receptor part. And that G protein is going to have GTP in it, and it's going to hydrolyze it to GDP, and it's going to go out and it's going to activate things and cause stuff to happen inside the cell. There's a whole signaling cascade that's downstream of this that's going to lead to proliferation of the macrophage or T cell, which means that by the time our virus has gone through all the process and actually got in there, this cell is going to get ready to divide, and it's going to divide with an embedded copy of HIV in its genome, which means there's now 2 HIV infected cells, and so HIV kind of wins by not doing anything, which is very much the HIV situation. I have no idea what we're watching on Vimeo, but whatever, yeah. As long as it doesn't turn to something terrible, we can just watch it. Here are some structures showing CCR5 with GP120 and CD4 in there. So CD4 binds to a different part of GP120. It binds that hydrophobic pocket. That causes the V3 loop to come out, and V3 loop is this sort of cyan thing that's sticking down into there, and that is going to have the alpha helix on it that's going to actually interact with and activate CCR5. Once CCR5 is activated, then CCR5, in addition to causing a signaling cascade to happen inside the cell, is going to cause clathrin-mediated endocytosis. This is the baby version. I'll leave it there for you because it's nice to be babied a little. Yeah, it's like a spa day for your brain. Let's go to the hard, real version, if you like. This is even pretty rounded off. So clathrin will interact with adapter proteins. The main one for clathrin-mediated endocytosis is going to be adapter protein 2, abbreviated as AP2. That's these little brown dudes that are going to be right there. So the AP2 is going to interact with the receptor when the receptor has bound to a ligand, and HIV is mimicking the natural ligand of CCR5, and so that happens. This is a mechanism that means that once your G protein coupled receptor gets its little, clamps down like a Venus flytrap on its ligand, then it has to be pulled in because it's going to start activating its signaling cascade. And that signaling cascade wouldn't ever necessarily stop until we physically destroy the signaling protein and the little thing that it bound. This is our off button. Our off button is we're going to destroy CCR5 and make a new one, because that's how cells work. And this is also going to be a control mechanism, so if you get signaling on one thing, we're going to pull it in. And so if we're going to do something, we got to wait for more signals to hit more places, and then, this is a way of sort of modulating the signaling that's going to happen. So clathrin interacts with itself. It makes these sort of trimeric cages. The trimeric cages are going to interact with the adapter proteins that are going to interact with the cargo proteins, and they're going to form these cages. The cages are going to form a nice little shell. The shell is going to recruit some actin polymerization. We're actually going to make cytoskeleton to sort of close this structure. So we're going to just build cytoplasm to push this close. And then there are bar domains and dynamin. So the bar domains are up here, and the dynamin is down here. F bar domains are going to regulate the size of the actual clathrin-coated vesicle, because clathrin can only assemble into cages of a certain size, which means that you gotta pinch it off. You gotta make sure that your vesicle is within the size range that clathrin is able to encapsulate it, if that's even the right word, to capture it. Yeah. And so then dynamin is going to be this little lasso that, snaps the neck here, and so that's actually going to break our little vesicle free. HIV is going to be where the green dot is right now, so it's going to be pulled into the cell, usually via clathrin-mediated endocytosis. And you'll be able to find experiments out there showing that there is variation. Certain strains aren't as good at this. Whatever. This is the general case as I would like to see it on the exam, and it's a reasonable, usual case scenario, I would say. Okay. The clathrin-coated pit, once it is pulled in, the clathrin is actually going to dissociate, and there's a whole process behind that. You can learn it or not, whatever. It's not virology, or I don't know. Everything connects to virology, and we got to draw a line somewhere. Now that's what we call an early endosome. So it's just a little vesicle that is free from the membrane, but it came from the plasma membrane. It's been folded in with an HIV inside it, in our case, and it's going to be moving into the cell. It's actually going to be dragged along the microtubules, and it's going to move by dynein. It's a little walking protein that's very good at walking things and dragging them toward the nucleus, basically. So that's where it's going to go. And that's normal, and normally we would drop this off at the lysosome, and it would get turned to acid and chopped up with proteases, and that would be great. That would be the end of HIV. But the way this is going to happen is we're going to merge this little tiny clathrin-coated pit after the clathrin falls off, so it's just a vesicle. We'll merge it with other vesicles. And there's a whole process that does that. There's cellular machinery. This is the normal process of endocytosis. Part of that machinery is going to be the vacuolar ATPase, which means it's an ATP-using enzyme that you find on vacuoles, vesicles, membrane enclosed bubbles of some sort. This thing is going to spin around, and it's actually a relative of the protein that makes ATP for us by pumping hydrogen out. In this case, we're going to spin the opposite way, I think. Pump hydrogen in. Yeah, we don't need that. And once we pump hydrogen into the vesicle, that's going to reduce the pH. We're going to have a lot of H+, little hydrogen ions floating around inside the vesicle. And we're going to spend ATP to do this. This is going to lower the pH of the vesicle, so it gets lower, 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 because when it gets to the lysosome, rather, that has a very low pH. It's like 2 or 3, something like that. And so we don't want to drop a big pH 7 blob of whatever from outside the cell into that, because it'll mess with the pH of the lysosome. And so we gradually acidify it so it's already down to lysosome pH effectively by the time it merges, or it's a lot closer. HIV would be destroyed if this thing completely acidified, but HIV has a get out of jail. It has an escape mechanism built in. The escape mechanism, we think, comes in the form of some histidines that are found in the GP41 region. There are several, they probably all do this. Histidine, under normal circumstances, is going to be neutral, which is to say, kind of hydrophobic. Yeah. Likes hanging around with other hydrophobic things like lysine, phenylalanine, leucine, all the things that just don't like water and like each other. It's like a little sort of focus group for hydrophobic things. That's what the inside of GP41 is like. When there's a lot of H+ around, the H+ will actually bind to this. One of these nitrogens will take on a hydrogen that did not have a hydrogen before. It's and it's going to use one of the electrons that it was sharing in here for that, and it's now grabbed itself a hydrogen because there's so much hydrogen around. It's like hydrogen pressure, if you like. That's going to happen at around a pKa of 6. So think like pH 6, 5.5 ish. Yeah, below 6 is where this is going to start to happen. That's called protonation. We're adding a proton to histidine, and it changes it into a positively charged ion, basically, version. That positively charged ion does not fit well with all of its former hydrophobic neighbors. And so that is going to drive a conformational change. And that's a big conformational change. This is going to be the last one. Part of that conformational change is going to involve spitting out the fusion peptide. Part of that conformational change is going to involve the heptad repeats. This is what heptad repeats are. An alpha helix, as we know it, of protein is going to make 2 complete turns. So like from there is 1, 2, like that. And it's going to do that every 7 amino acids. So amino acid 1 and 4, and then the one after number 7, like number 8, are going to be on the same side of the alpha helix. They'll all be like down or up or whatever. Their side chains will, rather. A heptad repeat is a thing we used to recognize when we were looking at proteins, and you can say, hey, if this was an alpha helix and we folded it up, look, all the gray ones here are hydrophobic, and those all end up on kind of one side of this alpha helix, and there's hardly any of these gray ones on the other side. This is a heptad repeat. It means every 7 amino acids there is a pattern in that 1 and 4 are going to be hydrophobic usually. Yeah. And sometimes it's 1, 3, and 4, or 1, 4, and 5, something like that. Yeah. 1, 2, and 4, whatever, like that. There are 2 heptad repeats in the GP41 part. And they're going to be important. And they are both amphipathic alpha helices. So you can say hydrophilic, likes water. That's like the white residues. You can say hydrophobic, hates water. But what we got, amphipathic means two-faced. It means it has one side that's hydrophobic and one side that's hydrophilic. There's no rules for like how big a side has to be. It could just be like one of these positions. Usually it's going to be like 2 or 3. Yeah. And that's going to create a hydrophobic face. The hydrophobic face is not going to be as stable around water. So this is going to drive an interaction where the hydrophobic faces are going to stick together. And that is going to drive one of our big conformational changes. Okay. The other part of the process is going to be down in the tail. So we're going to have this thing, the MPER. It's the membrane proximal region. It's folded up like this in this model. We think it actually sits in the membrane. So MPER would be sort of floating in the membrane, and then the transmembrane would be coming through. Now the transmembranes probably aren't as far apart as it's showing here, but it's like a schematic. They're just showing you, somebody's idea of it. The MPER has 2 important amino acids in it, which are a tryptophan, that's this W672, and a phenylalanine, which is again F673. Those are both aromatic, and those are both very hydrophobic. They're on, the MPER is an amphipathic helix. It's got a little kink in the middle of it where it's not a helix anymore for a sec. But this side's hydrophilic, this side is very hydrophobic, and so it sort of floats in the membrane. It's going to float half submerged in the membrane of the virion. And in this part, these 2 amino acids are actually going to interact with lipid molecules there in the membrane of the virus. That's going to be important for later. They're going to interact with that so tightly that if they get pulled out, if this whole helix gets pulled out of the membrane by something happening, then they're actually going to still, we think, be holding on to those lipid molecules. And that's going to help drive the fusion process, because you're going to get lipids that are not in the membrane, which is going to lead to other lipids sort of rearranging themselves to make that not as energetically unfavorable as it is. Yeah, because it is. Now, this is the same transmembrane as you saw here. Transmembrane goes through the membrane. As you can see, they are close together. They are not far apart like it shows here. That's goofball stuff, but it's schematic. This is what we think they look like, and the cytoplasmic domains are all of this part down here. So it's sort of 3 alpha helices, 2 little kinks in there. These alpha helices are going to form a sort of a flat disc. They'll sometimes draw it as like a little cylinder, and this is going to be on the bottom of the transmembrane part. These are palmitoylated, so they have palmitic acid attached to them, covalently attached to the protein. It's attached at 2 cysteine residues, 764 and 837. And those palmitates are going to poke back up into the membrane on the inside of the virion. Yeah. Or, inside of the plasma membrane before this thing buds. Yeah. Because the one is going to become the other. The plasma membrane is pinched off and becomes the virion. Okay. So palmitoylated, sort of basal disc, if you like, of the envelope protein. Here is the super broad summary. So you start out in the closed pre-triggered conformation. This is before it encounters CD4. Encountering CD4 causes that whole rippling cascade of conformational changes in the GP120 and the GP41 parts. That's going to take us to our partly open conformation. And then it's going to go to this completely open conformation that exposes GP41. GP41 is going to embed its fusion peptide in the membrane. And once that fusion peptide is embedded in the membrane, then heptad repeat 1 and heptad repeat 2, which is down here, are going to fold together. That's going to make the 6 helix bundle. And that is going to pull the MPER out of the membrane, and that is going to cause fusion. Now, I'll show you those last parts in a couple more steps here in just a moment. Yeah, Vimeo has died. Yeah, RIP Vimeo. Here is the entire mechanism. So we're going from closed to open conformation. We then bind the co-receptor. This is showing it coming apart. It may or may not come apart. But if it's in a reducing environment, that'll reduce the disulfide bond, and those would then potentially come apart. Or is it the other way around? I don't know. If it's the other way around, look it up. Yeah, that's fine. I'm not re-recording this. Then you get the conformational change where the fusion peptide, which is now an alpha helix because it was able to change shape, and it's now free because furin cleaved it, is able to go up and extend and embed itself in the cell. It's quite hydrophobic. And so this is the vesicle membrane. This is the viral membrane. You can see that basal disc here is the pink sort of cylinder at the bottom. Here's heptad repeat 1, which is right next to the fusion peptide. Then there's a little sort of looped out region here that interacts and doesn't really play a role. The heptad repeat 2 is shown down here. It's actually much shorter than heptad repeat 1. And now heptad repeat 2s are going to fold up on the outside. That's going to bring this membrane real close to this membrane. That's going to pull MPER out of the membrane. MPER is going to be holding a lipid molecule. And now that it's out of the membrane, it is going to cause hemifusion. So this lipid up here will attract other lipids to it. They'll be able to form with it. You will get a half fuse, so the outer leaflet of the vesicle will bind to the outer leaflet of the virion, and so those will be joined. They'll be fused as like one membrane, but the inner leaflets will still be independent. By leaflet, I mean the inner face of the lipid bilayer, and inner set of lipid molecules, if you like. And so after hemifusion, the 6 helix bundle, which is formed with like a lot of, as a big free energy step down in making that thing is locked in place. That thing does not reverse. And so now if you have at least 2, and it works better if you have more, of these envelope proteins that are all working together, then you can get from hemifusion, it basically can't go backwards, so it goes forward to full fusion. It is just the physics, as far as we know, that drives it to full fusion. But if you knock out the palmitic acids or mess around with this little base plate, then you cannot get past the 6 helix bundle stage. And so you cannot get to full fusion without those palmitates on the cysteines down there in the tail of the protein. So that's the kind of short version with all the details of the HIV entry story, and hopefully that'll help you once you've got the details down to prep for the exam question. And if you got this down, I hope you get that exam question. All right, see you later.